So we are very happy to see Laurentiu here. He will talk about structural bioinformatics and application of some data science machine learning methods based on the Gibbs sampling. So Laurentiu, please start your talk. Thank you very much for coming here.
So hi everyone. Thank you for having me. It's the first time on talking on this group. Is everybody
can you share the slides, please?
I didn't share the screen, did I?
Now, okay. Yes, now we see it. Okay, thank you.
Okay. So just briefly, I want to present my work related on molecular simulation. It's a new method that I've been working on for a little more than a decade, and it involves how to do molecular simulation a little bit faster. And the software that we use and we started developing a while ago, and we're still developing now, it's called RoboSample, because it's based on and uses algorithms borrowed from robotics. And it's meant to give us, or it's meant to be used for faster simulations and to get the conformational spaces a little bit faster. And now, why do we want this? First of all, why do we want this? The main problem that we are after, it's the macromolecular conformational spaces, which can get very big, very fast because of the dimensionality. And for example, well, during my PhD and afterwards, and even now, I've been working on a few projects of protein engineering, and mainly in plant biology, but also in other fields. And a lot of the times we are convinced that having a 3D structure of a macromolecule, of proteins in particular, because we dealt a lot with the proteins, and a lot of times we thought that having a 3D structure will help us and will do when we're trying to change the protein, and we'll try to get it to do what we want. But a lot of times that proved that it's not enough, and that the macromolecules are actually very flexible, and they can have a very large conformational space. Even if they're around some equilibrium conformation, they still have a lot of flexibility in it that if you don't account for, you usually don't get the results that you want. And I put a few examples here. I guess the first example is the spike protein, Sars-Cov-2, and I guess everybody knows that it's sitting like a tree or like a mushroom on the virus, and just trying to fish and trying to pick up the ligand. And this conformational these large conformational movements that it has, it was already studied by structural methods, and we can see that the movements are very large, and these kind of very large movements are difficult to get with, let's say, more established methods like molecular dynamics or Markov Chain Monte Carlo methods. And so that there's a big issue here, because most of the simulations that you can find online on databases involve a huge amount of computer resources and a lot of time to get the simulations, and even then you don't have this type of movement. You don't get to have this type of movement. So that's one example that I'm particularly interested in. It's proteins that have this kind of movements. Like, for example, another example is the antibodies that have two arms like this, and they are searching, right? They have a lot of movements in it, and they're usually just big domains that are bound by hinges regions. But this is not the only case that the conformational space can get particularly big. Another space that we can that we're after a lot of the times is the interaction between a receptor, protein receptor, or some other macromolecule receptor and the small ligand or peptide ligand. Why not? And I have this example here where we have a protein and its ligand, and we can say that the ligand is not really stuck there, it's not really fixed into one position, but it has a lot of positions. It has a certain degree of entropy, and we have to take that into consideration if we want to compute how tight those two are interacting. So in order to get the free energy of binding, for example, we want to get that entropy. We want to take a look at what's happening in the domain of the flexibility. Also, there are a lot of the proteins are intrinsically disordered proteins. They don't really have a conformation. They have a lot of conformations, possible conformations, and they only adopt the conformation once they get into their position. So what do we do with those? In order to sample them, you have a lot of degrees of freedom that you have to take into account now for real, because you don't have an equilibrium conformation that you know about, right? Like in the case of the spike protein. So that's basically what's the problem about. So what exactly did we do, and what is the flowchart for what we were thinking about when we developed this method? Let's say we have a molecular system and some sampling method that we want to use in order to get its conformational space down here. And this conformational space in particular is the conformational space represented in the two angles phi and psi of the peptidic bond, the smallest peptide model system that we use for sampling, and it's called alanine dipeptide. And it can be represented in a 2D surface. And as you can see here, the surface has a few minimals on the left and one minima on the right. And the problem lies in the fact that it's very hard to get from one minima to another. And so you have this molecular system, you have some sampling methods, so you want to do better for this. And so how does established methods, let's say classical methods, do this? You start representing your molecular system in some coordinates, some type of coordinates, generalized coordinates. There's a big pool of coordinates. And the simplest one are the Cartesian coordinates because they're not coupled and they can be used easily. And then you take a sampling method such as the molecular dynamics, which after you run it for a long time, it comes to resemble and to search the whole conformational space that you need. Or you can do a Markov Chain Monte Carlo methods where you can change the conformation little by little until you search the whole space. So what did we do? We said, okay, let's try some other type of coordinates that other people have tried it before, such as the internal coordinates representation. So what does that mean? It means taking the bond lengths of the molecule, the bond angles of the molecules, and the torsions of the molecules. This bond angle torsion system of representation is called internal coordinates. So instead of representing in Cartesian coordinates, we represented it in internal coordinates. And now, in these internal coordinates, let's try to impose some constraints. And the way to impose constraints, some constraints, is either by using maximal coordinates, where you have all your coordinates, all your dimensionality, and just add some new equations, usually in the form of Lagrange multipliers, and then solve the constraints. Or the other way that can be done is in reduced coordinates, which are fairly used in robotics, in robot mechanics, and they're the coordinates that represents the joints between different links in a robot, right? And the mechanical part of the robot is an articulated body. But okay, if there's still a problem here. Once you use constraints, you don't use all the degrees of freedom in your molecular system. So what should you do? You cannot just sample some molecule, but choose some degrees of freedom that you want to. Then you're not going ergodic, right? You're just using some subset of the coordinates. And this is where we thought that we should use Gibbs sampling, right? And Gibbs sampling is when you have a large multidimensional random variable, you sample by taking turns in the conditionals, right? And to be more specific, we, for example, first sample only the torsions in our molecules, and then having that conformation sampled, start from there, and then try some other set of coordinates, some other set of constraints, and so on and so forth. Some set of coordinates, another set, another set, another set, and in this way you go ergodic and you simulate all your degrees of freedom. The most popular scheme that we use is to sample all the coordinates or the internal coordinates, and then only the torsions, and then all the coordinates, and then the torsions, and so on. The way we do this in order to get the right probability distribution, which in the case of molecular systems is the Boltzmann distribution, it's by doing Markov Chain Monte Carlo. It's not like molecular dynamics. Molecular dynamics also, in the end, after simulating a long time, gets the distribution. But the Markov Chain Monte Carlo methods sample directly from the distribution. And but they have a downfall. They have, in the case of high dimensionality, and in particular in molecules, it's very hard to propose moves for the Monte Carlo method. So it's very hard to propose to just move an atom somewhere. It's very difficult. So one way to do it, developed in the 60s, yes, is the hybrid Monte Carlo method. So the move that you want to propose is by using molecular dynamics. And this way you just inject some kinetic energy into the system, and the system goes a long way, and it's more probable to jump off, on top of the energy barriers. And so this method, which combines hybrid Monte Carlo and constraints and Gibbs sampling, is just, we abbreviated GCHMC, and published in 2017. Now, it's all very nice, but we have to deal with some problems that the nice algorithms in robotics did not provide us. Usually in robotics, people deal with how to control the robot and how to do a path planning. Where in our case, we're not interested in that. We're just interested in random sampling, but we have to sample from the correct probability distribution. And usually the probability, it's represented here. It's just the exponential from a term that includes the temperature, and then there's the kinetic energy and the potential energy. Now, because the kinetic energy, as you know, is just the product between the momenta and the inverse mass, right? This is just the very well-known formula for the kinetic energy. But this mass matrix, in the case of Cartesian coordinates, is very simple. It's just a diagonal matrix that has masses on it. Now, in the case of generalized coordinates, it's not so simple anymore because the mass matrix has the masses on diagonal, but it's also multiplied by 2 Jacobians at each side. And so this mass matrix, to invert this mass matrix, it's difficult now, becomes difficult now. The algorithm, if you want to invert it directly, it's just not possible for systems that big. It's not feasible. It is possible, of course. It's not feasible. But again, robot mechanics algorithms already dealt with this, and now because it's a particular type of matrix, can be done in linear time. So we needed this inversion for the kinetic energy. Now, another term that we needed, it's again related to this matrix, and it comes because of the change of the coordinate system, right? So this is just correcting for the probability measure here. So you have the probability density, and then to correct for the measure, you have to add another determinant, right? So this determinant, which is also called the Fixman term, it's also computed based on the mass matrix, and it involves a determinant where this determinant is just as hard as the inversion of this matrix. So, but that it's solved already in the robotics, so we borrowed from there too. Just we had to adapt some of our libraries, and that was all. And so we kind of modeled our molecules as articulated bodies connected by joints as robots, and then did the simulation using the Gibbs sampling. We had some new improvements. Well, people have been doing molecular dynamics using robotic joints before, but they only use the torsional dynamics because it is known that the torsions in the molecules are doing most of the movements, but it's not enough. For example, in our model system, alanine dipeptide, we have 2 big atoms that cannot cross. They cannot pass one from another. You cannot pass one from another because of their steric hindrance. And so you need an angle 2 in order for those to do their conformational change. So only doing torsional dynamics, you'll never get the whole space. In the case of proteins, this becomes a lot worse. So only using torsions, it's really not an option. So you do Gibbs sampling, and that's one way. And another way is to use some more complicated joints that we introduced recently. So for example, we introduced the cylindrical joint, which uses a torsion, but it also uses prismatic type of move. Or we use some other type of joint, which is like the knee joint, right? Which uses, it's called a ball in robotic terms. And it just has 3 rotational degrees of freedom now. So, okay, you don't allow any bond lengths to move, but you allow all the angles and all the torsional angles. So one other thing that we're using now, it's the rigid body dynamics. So to be a little bit more clear, the way we do Gibbs sampling is by alternating fully flexible system with only torsional system molecular dynamics in between the exception rejection step of a hybrid Monte Carlo protocol. But now we can introduce some bigger bodies of the entire molecular system. So not all the torsions in the molecule, but just keep fixed two bodies of the molecules and only allow several torsions, for example, to move, like in the case of the spike protein or in the case of an antibody. And so this improved our method a little bit, and have several examples of our results, both published and a few not yet published, but a case that we demonstrated that the method is better than regular Hamiltonian Monte Carlo or hybrid Monte Carlo is the case of the ligands that contain macrocycles. And this type of ligands are notoriously difficult to sample because they have these large cycles, and these cycles just don't get from one conformation to another very easily. Another case that we showed that we get a better sample in the same amount of time, in the same amount of computer time. It was the case of glycans that are bound to proteins. Yet another case, these are still to be published, but yet another case are big domains of proteins that move in relation to another. And I have this protein here that has three subdomains, and I can move the three subdomains freely. Yet another case that I've been playing with is just the interaction between a ligand and a receptor within a ligand binding site, which is a very nice example. I'll be pursuing it further. And to get numerical with this, here's the numerical estimation of the efficiency in the case of 5 macrocycles. And in what I have I have two main curves, one representing the number of clusters as it grows with times, and the number of clusters, it gets bigger with GCHMC than with regular hybrid Monte Carlo. It gets bigger faster. So just this way of getting the efficiency of now just counting the clusters that you get once you simulate a molecule. Another way to quantify this, to quantify the success of the method was to represent the root mean squared deviation of the structure as the time went on, as the time of the simulation time went on. And then, of course, the frequency that it crosses the energy barriers here in the middle. The middle of this chart is the energy barrier that our molecule has to cross. And so we showed that it's 10 times faster. And yeah, that's it. We're still working on this. It's a lot of work to be done yet. And yeah, we're doing this with the Illinois Institute of Technology. And yeah, I guess that's it. Thank you.
Thank you very much. Thank you very much. So colleagues, please feel free to ask questions. So meanwhile, let me ask you questions. So the fact that you said that it is difficult to invert this matrix M of Q. So can you please repeat how you resolve this issue?
Yeah.
Yeah, this.
We're talking about this matrix, right? This
Yeah.
matrix is just, it contains a Jacobian of the transform between the type of coordinates, between the Cartesian coordinates and the internal coordinates, for example, or other type of reduced coordinates. Now, this matrix has a special structure, and the mathematical details are, I guess I need an hour just to go through them. But I guess it has a special structure because it embeds in it all the information about how the bodies are linked. So once you know how the bodies are linked and all the vectors and what degrees of freedom they have, and you know the masses and the inertias that they have, you can deduce an algorithm that can do this recursively. That's all it does. So it does not treat it as a regular matrix where you don't know anything about it and you have to invert it. Well, here you know about it. You don't even represent it as a matrix.
Okay.
There's just a compact way of writing it.
Okay. So about this, okay. Are you using some special software, or you develop it by yourself? Or what about two things, about the Gibbs sampling and about this inversion of the matrix?
So we wrote the Gibbs sampling part. We wrote the hybrid Monte Carlo part in generalized coordinates. We adopted a lot of the molecular modeling for ourselves. And we are based on libraries from robot mechanics. We only have to just modify a little bit from that libraries to adapt to our molecular systems. So there were no new, let's say, new developments in terms of robot mechanics algorithms or in terms of molecular modeling, not very much. We did developments in the new joint area where, from what I know, nobody looked at before. And then the sampling part is ours. Yeah, we thought about just how to mix this type of sampling to get the ergodicity, to get all the degrees of freedom. And yes, it is based on some libraries, and we developed a library too. Yeah.
Okay. What kind of computational? So you have some clusters, or it's less heavy, or it's how much computational power are you using?
Oh, yeah, that was the idea, just not to reduce the computational power. I'm doing on my laptop a lot of the simulations.
Oh, okay, great.
Yeah, well, if you want to get the, well, you can use like desktop computers for several days for one molecule, like medium-sized protein, let's say. Or it depends on what if you go after free energy calculations, you may want to go further with this. Yeah, but it's pretty difficult. This is the first thing that we had in mind, the free energy calculations, which you can go for months with those. Right. Yeah, and that's because of the sampling. That's because of the sampling problem of the receptor and the ligand. So, and the nice thing about it is that you can couple it with another method. It's independent. So I already coupled it. Already working with replica exchange, for example. So yeah.
Thanks for the talk. It was very interesting. I wonder on the graph on the plot with the number of clusters, why it does not converge? Because I saw that it was like increasing all the time. Did you achieve the convergence, at least for some systems, like the plateau in the number of clusters? Or is it because, you just continue sample and sample very large conformational space?
Well, for most of them, it looks to me like for here first.
Yeah, sorry, I missed that one. Yeah.
And then the last one was close. what I did was I wanted to show here not necessarily the convergence of the simulation, which is almost converged in most of the cases, but that was not the main idea. The main idea was to show that it gets more cluster more rapidly than regular HMC to compare the curves.
Okay, thanks.
Sure.
Colleagues, please don't hesitate to ask questions.
I also have another question. If I'm right, you now use like almost atomic precision. So you introduce joints for like normal torsion angles. Yeah, did you try some maybe coarse-grained implementations, like more coarse-grained, like at least several amino acids maybe per particle or some, or at least something like Martini level maybe?
Yeah, we can use rigid bodies of that size or size that is even bigger than that. The idea was not to lose any accuracy in our simulations. That's why we wanted to use the Gibbs sampling method and not to reduce the level of detail of the simulation. Why? Because the whole group was involved in free energy calculations, and in the free energy calculations, the level of accuracy is critical. You don't want to go and use coarse graining in the free energy calculations. Quite the opposite. You want to use all the atomic detail that you need. And yeah, being on that line, we wanted to use some method that it's ergodic, it's going after all the degrees of freedom, and it's not making any compromise in some way. So, but it can be coupled with these kind of simulations by doing this sort of simulation just to show you some trends that you can use in order to establish your parameters, I guess.
Okay, because, you know, from the efficiency of sampling, or like from the speed of sampling, it seems it can be very useful for cryo-EM when you need, you know, like for a lot of different starting conformations to somehow integrate them with the all the cryo-EM data or integrative modeling and so on. That's why I was interested.
Yeah, indeed. Yes, indeed. Yeah, it can be useful. We didn't collaborate with any group of cryo-EM, but yes, definitely can be useful here.
Okay, thanks.
Sure. Yeah.
Okay, so maybe a naive question about maybe also for Artur. Have you thought about relating this with this Alphafault, which is now kind of big hype? Everybody's discussing Alphafault.
No, I didn't try to. No. Yeah, we are using Alphafault in our group. No, I didn't try to integrate anything with the modeling part. I am very interested in the folding, but I didn't do any real folding with GCHMC yet. But yeah, it would be very interesting to do, sure. It's one of my, let's say, fun thing to do, because it's very difficult, right? So.
Artur Zalewski, who asked you the previous question, he is expert in the Alphafault. And so as far as I understand, his question about this cryo-EM may be related to the, as far as I understand, Artur, this, what you mentioned, this is the case where Alphafault is not working somewhat very good, yes, cryo-EM, am I right?
Not, in fact, because all biological molecules, they are flexible, and Lorenz's talk just demonstrate all the degrees of this flexibility. So usually in cryo-EM you have like a mix of multiple states, really different conformations. So to properly understand them, you usually generate like an ensemble, computational ensemble, but Alphafault does not give you like a lot of freedom there. And with Lorenz's approach, it can be extended. So you can use Alphafault structure as input for this approach to generate more structures, and after that feed them in the cryo-EM data or any other integrative modeling, like with SACS data, with cross-linking chemical, and so on and so on. Because now we do not have many information or robust ways to, of course, molecular dynamic simulations with enhanced sampling, it's very, very powerful approach, but it's still very, very expensive. Yeah, and this kind of approach can give a lot of information, very useful. Yeah.
I did compare it with some NMR models. I didn't compare with any cryo-EM, but it's a very good idea. But I don't know if you can see my screen.
Yes,
yeah.
My picture, yeah. So I did a simulation, short few hours simulation with RoboSample, and looked at the ensemble of models from NMR, and it looks similar. So yeah, I used it in comparing with that experimental data. But I guess with cryo-EM would be, it's a very good idea.
Oh, and so am I right that you can apply custom constraints in your approach, not just, those which comes from like distances, angles, and so on? I mean, like just, custom constraints, like this domain and this domain should be not further away than like X angstroms and so on. So, and does it affect, like sampling efficiency or performance at all?
Well, the more constraint, the more.
I mean, like very distant constraints, like when you have two very distant parts of the proteins, not some, like local constraints.
Right, I can use specific constraints, like by adding equations with Lagrange multipliers, which are, so these are on top of the simulation, just using, so you can add new equations to solve in order to keep the constraints, that are implemented in generalized coordinates. So they do. Well, I'm not doing that. There's this library of robotics that are doing it. So they're just using the Jacobian of the transform between the coordinates, and they can impose constraints really easily. Yeah.
Okay, great. Thanks again.
Sure.
Sasha, Pavel, did you have some question?
Yes, do you hear me? I tried to switch on the mic.
Yes, we hear you.
It's actually it's now it's not a question. It's kind of like refining of what Artur asked, but it's a bit more general. Did I understand correctly that you suppose that the Markov chain of the structures eventually, if it like run a long time, it give you the distribution of the possible conformations which represent the real world distribution. Not only the best solution is the same, the whole distribution is more or less the same.
Right.
It's really great. Yeah, thank you, because it's much more than to know the maximum. Okay.
Oh, yeah. It just draws samples from the distribution. Yeah. So I'm hopefully that's the idea, to hopefully get all the minima and all the everything. Yeah, to get everything.
Yeah, actually, it means that you can, if you have any function on the structure, you can calculate its momentum on the distribution you generate from your Markov chain, and it is a good estimation of the real distribution of this function in real world.
Yeah, that's what I'm trying to do. Yeah.
Yeah, it's thank you for
Sure.
very good story.
Okay, colleagues, please ask questions. Or if not questions, let us thank Laurentiu. Thank you very much.
Thank you too. Thank you for having me here, first of all.
Thank you very much. Okay, so let me stop the recording.